Cancer. It's a word that strikes fear into the hearts of millions. But what if I told you that the future of cancer treatment is filled with hope? We're going to explore the top 3 cutting-edge cancer therapies that are revolutionizing the fight against this deadly disease. Number 1. CAR T-cell therapy. First on our list is chimeric antigen receptor T-cell therapy, CAR T-cell therapy for short. This revolutionary treatment has been making waves since its FDA approval in 2017 for certain types of blood cancers. CAR T-cell therapy is a type of immunotherapy that uses your body's own immune system to fight cancer, specifically your T-cells. The process begins by collecting a sample of your blood, from which scientists can extract your T-cells. These cells are then genetically modified in a lab to express chimeric antigen receptors on their surface. CARs are synthetic proteins designed to recognize and bind to specific antigens on cancer cells. This genetic engineering process essentially turns your T-cells into cancer-seeking missiles. Once the CAR T-cells are ready, they are multiplied in the lab until there are millions of them. Before retrieving the CAR T-cells, you may undergo lymphodepleting chemotherapy to make room for your new cells. The CAR T-cells are then infused back into your body, where they continue to multiply and patrol for cancer cells expressing the target antigen. When a CAR T-cell encounters a cancer cell, it latches onto the antigen and activates, releasing cytokines and other molecules that help kill the cancer cell. The CAR T-cell continues to multiply in your body, providing a long-lasting, living drug that can potentially keep cancer at bay for an extended period. So far, CAR T-cell therapy has shown remarkable success in treating certain types of leukemia and lymphoma, with remission rates as high as 70 to 80% in some cases. However, it's not without risks. The most common side effect includes cytokine release syndrome, which can cause fever, low blood pressure, respiratory distress, and neurological toxicities like confusion, seizures, and encephalopathy. These side effects can be severe but usually manageable with proper care. Currently, 6 CAR T-cell therapies have received FDA approval for this treatment of various blood cancers, including lymphomas, leukemias, and multiple myeloma. Researchers are working to expand the use of CAR T-cell therapy to other types of cancers, including solid tumors. While challenges remain, such as tumor heterogeneity and immunosuppressive tumor microenvironments, the field is rapidly evolving, with new strategies being developed to overcome these obstacles. The incredible potential of CAR T-cell therapy lies in its ability to harness the power of your own immune system to fight cancer. By giving your T-cells the tools they need to recognize and attack cancer cells specifically, this personalized approach offers new hope for patients who have exhausted other treatment options. Number 2. mRNA cancer vaccines. A promising new approach inspired by the success of the mRNA technology in COVID-19 vaccines. While still in the early stages of development, these vaccines have potential to revolutionize cancer prevention and treatment by training your immune system to recognize and attack cancer cells. The core concept behind mRNA cancer vaccines is to deliver genetic instructions, mRNA coding for specific cancer antigens, into your cells. Once inside, your cells use this mRNA to produce the cancer antigens, which are then presented on their surface. Your immune system recognizes these antigens as foreign and mounts a targeted response, generating T-cells and antibodies designed to seek out and destroy cancer cells, bearing those specific proteins. One key advantage of mRNA vaccines is their potential for personalization. By analyzing a patient's unique tumor profile, scientists can identify the most immunogenic antigens, those most likely to elicit a strong immune response. This information is used to design a tailored mRNA vaccine encoding multiple patients' specific antigens, effectively training the immune system to recognize and attack that individual's cancer. The development of mRNA cancer vaccines has been accelerated by the advances in lipid nanoparticle technology, which allows for efficient delivery of mRNA into cells. These nanoparticles are engineered to protect the mRNA from degradation and facilitate its uptake by immune cells. Some researchers are even exploring the use of multi-layered nanoparticles that can carry larger amounts of mRNA, potentially enhancing the immune response. Early clinical trials of mRNA cancer vaccines have shown promising results in several cancer types. However, specific challenges remain in developing effective mRNA cancer vaccines. One major hurdle is the immunosuppressive tumor and microenvironment, which can dampen the immune response elicited by the vaccine. Researchers are exploring combination strategies, such as using checkpoint inhibitors or immunotherapies alongside the mRNA vaccine to overcome the immunosuppression and enhance the anti-tumor response. Another challenge is identifying the most effective antigens to target. While personalized vaccines offer the potential for highly specific immune response, they are also more complex and time-consuming to develop. Some researchers are investigating the use of shared tumor antigens, which are expressed by multiple patients' tumors, as a more streamlined approach. Despite these challenges, the field of mRNA cancer vaccines is rapidly advancing, with numerous clinical trials underway in various cancer types. The potential for these vaccines to not only treat existing cancers but also prevent their development in high-risk individuals is particularly exciting. Number 3. Radiopharmaceuticals, a class of drugs that deliver targeted radiation directly to cancer cells, while minimizing damage to surrounding healthy tissue. This precision approach represents a significant advancement over traditional external beam radiation therapy, which can cause significant side effects due to off-target radiation exposure. Radiopharmaceuticals consist of two main components: a radioactive isotope and a targeting molecule. The radioactive isotope emits radiation that can kill cancer cells, while the targeting molecule is designed to bind specifically to receptors or antigens expressed by the tumor cells. This allows the radiopharmaceutical to deliver a high dose of radiation directly to the tumor while sparing healthy tissue. One of the most promising radiopharmaceuticals is lutetium-177-PSMA-617, Lu-PSMA for short, which targets the prostate-specific membrane antigen PSMA expressed by most prostate cancer cells. Another example is radium-223 dichloride, Zovigo for short, a radiopharmaceutical approved for treatment of prostate cancer that is spread to the bones. Radium-223 is an alpha emitter that mimics calcium, allowing it to be taken up preferentially by areas of active bone turnover, such as bone metastases. By selectively targeting these metastases, radium-223 can alleviate pain, improve quality of life, and extend survival in some patients. The development of radiopharmaceuticals involves a multidisciplinary approach, combining expertise in nuclear medicine, radiochemistry, and oncology. One key aspect is the selection of an appropriate radioactive isotope. Different isotopes emit different types of radiation, alpha, beta, or gamma, and have different half-lives, which can impact their therapeutic efficacy and safety profile. Researchers must also design a targeting molecule that can effectively and specifically deliver the isotope to the tumor cells. Radiopharmaceuticals offer several advantages over traditional radiation therapy. By delivering radiation directly to the tumor, they can achieve higher doses while minimizing exposure to surrounding tissues. This can lead to improved efficiency and reduced side effects. Additionally, because radiopharmaceuticals are systemic therapies, they have potential to target metastatic lesions throughout the body, including those that may be too small to detect with imaging. However, radiopharmaceuticals also face challenges. One issue is the potential for off-targeting effects, particularly in organs that express the target antigens or have high blood flow, such as the liver or kidneys. Careful dosing and patient selection can help mitigate these risks. Despite these challenges, the field of radiopharmaceuticals is rapidly expanding, with numerous agents in clinical development for a variety of cancer types. In addition to prostate cancer, radiopharmaceuticals are being investigated in neuroendocrine tumors, lymphomas, and breast cancer, among others. As our understanding of tumor biology and radiochemistry advances, we can expect to see more targeted and effective radiopharmaceuticals emerge, offering new treatment options for patients with advanced or refractory cancers. But this is just the beginning. With continued research and support, who knows what incredible breakthroughs tomorrow may bring? Let us know your thoughts in the comments below. What excites you most about cutting-edge cancer treatments? If you're not already subscribed, hit the subscribe button to stay up to date on the latest development in cancer research and other fascinating science topics. Thank you for watching, and we'll see you next time.